Hello, welcome. My name is Jennifer Griffith, and I work at NMOA, which is the Northeast Waste Management Officials Association. So I've got the webinar up on the screen. Jennifer, are you seeing the webinar? I mean,
yes, the website. Yes, I can see it.
So just so people can get a better feel for what NMOA is, if you aren't already, already aware, we're an association of the 8 Northeast states. We were formed by the governors of the New England states back in the 1980s to focus on waste issues, and the wayside cleanup programs are also members of NMOA. And then New York and New Jersey also joined. So we are the 8 Northeast states. And the wayside cleanup programs in the states, one of their top priorities for NMOA is information sharing and training for their staff. So that is what we are doing today. Is PFAS is a big topic and has been since about 2016 with the states. So we are continuing our webinars on PFAS, and we have a whole series. We've got today's webinar and 9 more scheduled. So part of the reason why I am showing the NMOA website is also so that I can show you afterwards. The presentations will be posted and where you'll be able to access them. So here's just NMOA.org. We have a very old website. It's built back in the 90s, I think. But over here is Wayside Cleanup. You click on that, then you get to Wayside Cleanup. You can click on Events. And the one out furthest in the future is at the top. We have a couple of conferences that we were going to hold in 2020 that we had to postpone. But then also here are all the webinars that are coming up, and they're going from the furthest one to the most recent one. So here, when you scroll down, you'll get to past events, and this is where you'll be able to find today's slides. They'll be posted. The webinar we had last week is here, and then we've done a lot of other wayside cleanup related webinars. We did a series of 5 that were not PFAS, and then we've also done last summer a whole bunch more PFAS webinars. So and we've done workshops. We usually do in-person workshops on different topics, so we have a whole lot of different resources here and presentations that you can utilize. So I just wanted to show everybody that. So we have a lot of people joining us today, so we will have to do question and answers through, questions through typing. So please, at any point, you don't have to wait till the presenter is finished to be able to type in your question. You can type in questions at any point, and after the presenter is finished, I will ask questions of that presenter. So I think that's most of what I have to say. I'm going to add a little disclaimer here that this webinar is for informational purposes and to stimulate discussion. You should not, does not mean that this is the position of the NMOA states or of NMOA. So I think I'll leave it with that. And so our first presenter, so today we're talking about PFAS and health effects, and specifically looking at mixtures, how you're not necessarily exposed to one chemical at a time. So we're going to explore that further, and we've got 2 researchers from Boston University School of Public Health to present today. So I will introduce the first presenter and turn. We're going to do it a little bit opposite of how it was originally written on the webpage. Dr. Jennifer Schlesinger is going to go first. She is a molecular toxicologist and associate professor of environmental health at Boston University School of Public Health. The overarching goal of her research is to determine how exposure to environmental toxicants and consumer product chemicals impairs bone, liver, and adipose homeostasis. And I learned that adipose is fat, for those of you that aren't steeped in all this. Specifically, she investigates nuclear receptor activation and how they affect bone, fat, and liver. The chemical classes she studies spans organotins, phthalates, organophosphate esters, brominated flame retardants, and of course, PFAS, which we're going to be hearing about today. So with that, take it over, Jennifer.
Hello, it is my doctoral student, Graylin Nielsen's, and my pleasure to be speaking with you today. This audience well knows that people are exposed to mixtures of PFAS in food, water, air, and dust, and that we need better biological systems to examine mechanisms of action that lead to adverse health effects that are observed in humans. So why do we need to know how things work? Well, because by knowing mechanisms of action, we can intervene and mitigate adverse health effects. Knowing mechanisms of action is also essential to being able to efficiently model and predict the biological and toxicological outcomes of exposures to chemical mixtures. So I'm going to begin with mechanisms of action side, followed by Ms. Nielsen discussing how we approach modeling the toxic effects of mixtures, including PFAS mixtures. Today, I'm going to share our investigation of the effects of PFOA, perfluorooctanoic acid, on liver and serum triglyceride and cholesterol homeostasis in a humanized PPARa alpha mouse model that's been fed an American diet. And an underlying theme of what I'm talking about today is how do we design studies in animal models to test and to understand the associations observed in human epidemiology. So as I said, this audience knows well that PFAS are a large group of chemicals used in consumer products, industrial processes, and firefighting foam. And as such, we are typically exposed to mixtures of these chemicals, which Ms. Nielsen is going to address more thoroughly. Another important consideration when studying PFAS is their unusual chemistry. So PFAS are not lipophilic. They don't dissolve in fats like the majority of environmental contaminants. Actually, they are amphipathic. What I mean by that is that they have a charged end and they have a neutral end. And because of this leads to their binding to serum proteins. So much of the PFAS in our body are bound to our serum proteins and are found in our blood. This chemistry also means that these chemicals don't move easily through membranes by diffusion. They require transport by proteins. In humans, serum PFAS concentrations are associated with adverse birth outcomes, reduced response to vaccines, reduced bone quality, as well as increased serum cholesterol. So why do we care that PFAS are associated with an increase in serum cholesterol? Well, that's because atherogenic dyslipidemia, which means elevated triglycerides and decreased high-density lipoprotein-associated cholesterol, and increased blood LDL, or low-density lipoprotein-associated cholesterol, are major contributors to cardiovascular disease, which is the leading cause of mortality in the United States and the Western world. So regulatory agencies use epidemiological endpoints as well as animal model data to establish safe exposure limits. In 2018, the European Food Safety Authority found that considerable portions of the European population exceeded the tolerable weekly intake for both PFOA and PFAS. The point of departure that these tolerable weekly intakes were based on was determined based on increased total serum cholesterol, as well as reduced vaccine response for PFAS. And this was all based on the human epidemiological data. So this just underscores the importance of the association between serum PFAS and the increase in serum cholesterol. However, in 2020, EFSA updated the critical endpoint for their safe exposure limit calculations. Based on available studies in animals and humans, effects on the immune system were considered the most critical for risk assessment in the 2020 update. So why the change in the endpoint for determining the point of departure? Actually, there were two issues that were raised. First, there were questions about whether the epidemiological association was a result of physiological confounding. That means that the serum PFAS concentrations and serum cholesterol concentrations were changing together because another variable was affecting them both, rather than PFAS were causing the change in serum cholesterol. The second issue was that some rodent models did not show the same increase in serum lipids and cholesterol. So just notice here in this justification of changing the critical endpoint based on available studies in animals and humans. Well, some argued that the lack of being able to reproduce the effect of PFAS on serum cholesterol in a rodent model suggested a lack of causality. So following from that second issue, do rodent studies imply the epidemiology studies are incorrect? So for example, Power et al. Is one of a number of studies that have found decreased serum cholesterol and triglycerides in mice given high doses of PFOA. I'll come back to this in a moment. Human relevance is reflected in multiple aspects of the experimental design. So first, we have to think about, was the dose, and in the case of PFAS, the serum levels that were generated in the study relevant to humans? Was the diet that was used relevant to humans? Diet is a very important source of cholesterol and also a very important contributor to regulation of serum cholesterol. We also have to recognize that even within rodents, not just not between rodents and humans, but even within rodents, there are strain differences in responses to PFAS. And finally, there are potentially important species differences to consider. So first of all, rodents do not express a protein called CETP. This protein transfers cholesterol esters from high-density lipoprotein cholesterol to low-density lipoprotein cholesterol, and triglycerides from low LDL to HDL. Power et al. Argues that it's necessary to have a model with a human-like cholesterol particle profile in order to understand the effects of PFAS. And so you'll see they use a very complicated transgenic mouse model. Another very important species difference has to do with PPARa. And as you'll see, I'm going to argue that actually what's necessary is to have a model that expresses a human PPARa in order to understand the effects that PFAS have on serum lipids. So in my lab, our goal is to develop human relevant animal models, and in this case, to test the hypothesis that PFAS induce dyslipidemia through human PPARa activation. So one way we can visualize the connection between PFAS, PPARa, and dyslipidemia is to construct something called an adverse outcome pathway, also known as an AOP. An AOP is a model that identifies the sequence of molecular and cellular events required to produce a toxic effect when an organism is exposed to a toxic agent. An AOP begins with defining the chemical properties of the agent. In the case of PFAS, PFAS are not readily metabolized, and so the bad actor is the parent compound, is the PFAS itself. In addition, the amphipathic nature of PFAS is going to influence which organs the PFAS reach and which organs they can actually enter. Next in our AOP is the molecular initiating event, which is the interaction of the agent, in our case, PFAS, with a macromolecule that begins the toxic process. In our hypothesis, PFAS initiate the toxic process by binding to and activating peroxisome proliferator activated receptor alpha. That's the only time I'm going to say that, also known as PPARa. Next in our AOP are the key events. These will be not only organ, in our case liver, but cell type, in our case hepatocyte specific. Early events include changes in signaling processes or protein expression that occur at the cell level. Late events include altered organ or tissue function. So PFAS, through their activation of PPARa, alter lipid homeostasis pathways in hepatocytes by changing which genes are being expressed, which leads to disruption of cholesterol and or triglyceride processing in the liver. The culmination of the AOP is the adverse outcome, which results from the organ dysfunction. And in our hypothesis, PFAS induce adverse outcomes such as increasing serum VLDL-C, which is the particle that carries triglycerides, and LDL-C, low density lipoprotein cholesterol, which remember are major contributors to cardiovascular disease. So before we get to our experimental model and data, I want to introduce you to a couple of important biological processes. So first, you may have noticed that I've been lumping together serum triglycerides and cholesterol. Why am I doing that? Well, because cholesterol is packaged in particles with triglycerides in the liver. So if you didn't know it, this is a liver here on the left. And there are two sources of fatty acids and cholesterol molecules to the liver. So why am I mentioning fatty acids all of a sudden? Fatty acids are the building blocks to form those triglycerides I've been talking about. So the liver receives fatty acids and cholesterol from food. It also has the ability to generate, to synthesize, to make fatty acids, which are used to make triglycerides, and to make cholesterol. The cholesterol and triglycerides then leave the liver packaged in these VLDL particles. Why do they need to be packaged? Because they don't dissolve in water. And guess what? Your blood's made of water. So what you see here on the right is a VLDL particle, a very low density lipoprotein particle. So you see on the inside of this particle, you have cholesterol, the molecule cholesterol, and triglycerides. You'll see that they're surrounded by the single-sided membrane that has some proteins that are embedded in it. And so this particle leaves the liver and travels around the bloodstream, and the triglycerides are released so they can be used by tissues. So what happens is that this particle then becomes smaller and denser. And so it becomes the LDL particle, the low density lipoprotein particle. And it's this LDL particle that delivers cholesterol to cells. Now we've also got this other type of cholesterol particle that you're probably familiar with, which is HDL, or high density lipoprotein cholesterol particle. The job of this particular particle is specifically to bring cholesterol back to the liver. So another important process here is that both LDL-C and HDL-C are bringing unused cholesterol back to the liver to be processed, reprocessed, or removed from the body. So how do we detect and measure these different types of cholesterol particles? Well, we collect blood, could be from a human, could be from a mouse, and we separate the particles based on their density. Then we measure the triglycerides or the cholesterol in each fraction. So what you see here is a representation of the typical distribution of triglycerides across the different types of cholesterol particles. So lower density is here on the left, higher density is here on the right. And what you see in a human is that the vast majority of triglycerides are carried in the VLDL particle, although some are carried in the LDL particles as well. Almost none is carried in the HDL. What does the cholesterol distribution look like? So in humans, the vast majority of cholesterol is carried in these LDL or low density lipoprotein cholesterol particles. Although you still see some that's being carried in VLDL particles and some that's being carried in HDL particles. One of our challenges in studying changes in blood lipid homeostasis is that there are different distributions of triglycerides and cholesterol across these particles in mice and rats versus humans. And this is for two reasons. As I said before, rodents don't express that protein CETP, which moves cholesterol from HDL, so in a human from HDL to LDL. And uptake of LDL by the LDL receptor is in the liver occurs much more rapidly in rodents than it does in humans. So what does this mean for the distribution of their triglycerides and cholesterol? So what you see is that in the mouse, again, the vast majority of triglycerides are carried in these VLDL particles, and even less than in the human is carried in the other types of cholesterol particles. When we look at the distribution of cholesterol, you see the vast majority of cholesterol is carried in HDL particles in the mouse, with the low density lipoprotein particles, the cholesterol that they're carrying is just showing up as kind of this shoulder on the HDL particle peak. So the second thing I want to introduce you to is PPARa. PPARa is a protein. It's very highly expressed in liver and in other organs that have a high energy demand, like the heart. PPARa is particularly important during starvation or endurance exercise because it allows the body to release energy stored as lipids. But in humans with PPARa mutations, so differences in their DNA sequence, and in mice in which PPARa has been knocked out, so like the mice don't express the protein, we see changes in serum cholesterol and triglyceride under normal conditions, suggesting that PPARa has an important role to play in regulating lipids under everyday conditions. So PPARa is a protein that belongs to a class of proteins called nuclear receptors. These types of proteins, these nuclear receptors, are the mechanism through which hormones and hormone-like signaling molecules change what genes are expressed and therefore what proteins are expressed. So when PFAS bind to PPARa, PPARa binds to DNA and either increases or decreases the expression of particular genes, which leads to changes in protein expression. So PPARa, in the liver in particular, controls the expression of a wide range of proteins involved in lipid uptake, synthesis, transport, storage, oxidation, and excretion. So PPARa has been at the heart of controversies over PFAS-induced biological effects. So what is the big stink about PPARa? So number one, there are species differences in the way PPARa is expressed and acts. So human PPARa is expressed at a lower level, meaning like if you compared the same amount of mouse liver to same amount of human liver, the mouse liver would have more PPARa in it than the human liver. Human liver is thought to be less responsive to activation than rodent PPARa. Rodent PPARa induces liver cancer, but human PPARa does not. So these have all been used as arguments to suggest that PFAS cannot act through human PPARa and that all rodent data on PFAS are irrelevant to understanding effects on human health. The second issue is that PPARa is a therapeutic target in the treatment of high blood lipid levels and high triglyceride levels. And in fact, fibrate drugs are prescribed to reduce serum triglycerides. So this has been used to argue that it cannot be possible for PFAS to increase serum lipids. And our experimental approach is designed to tackle these issues. So how did we test the hypothesis that PFAS induce dyslipidemia through human PPARa activation? So I'm going to begin with our mouse models. We actually used two mouse models. In the first of which, the PPARa gene has been knocked out. That means that this mouse cannot make PPARa at all. Our second model is a mouse which did not express PPARa. We've introduced the human gene, the full human gene, into the mouse, so that human and only human PPARa is expressed in the same organs and at the same level as it is in humans in our mouse. Another important aspect of our model is that we used both female and male mice. To date, there is only one other paper that has reported results of effects of PFAS on serum lipids in a female model. So our mice were treated with control water or PFOA containing water. This was in their drinking water. And at the rate at which they drank the water, we estimate their daily exposure to be 0.7 milligrams per kilogram per day. The mice were young mice. They were treated for 6 weeks from the age of 3 to 9 weeks. And then the second humanized variable in our model was the study diet. So we designed the diet for these mice to have the same macro and micronutrient distribution as in the NHANES What We Eat in America analysis. Because we were using young mice, we actually designed the diet based on the analysis in 2 to 19-year-olds. So it actually is a little high in sugar, 24%, 51% carbohydrates overall, 33.5% fat in the form of soybean oil, lard, and butter. And the reason why butter is important is because it also contains cholesterol, which is something, so fat and cholesterol are rarely significant parts of rodent diets. The diet had 14.7% protein, and we also made sure that the diet had a human-like contribution of both soluble and insoluble fiber. In order to confirm that perfluorooctanoic acid, which was the specific PFAS that we were working with in this experiment, so in order to confirm that PFOA was absorbed from the drinking water, and to be able to relate our results to humans, we measured the PFOA level concentration in serum. So first of all, notice there's no difference in the serum concentrations of PFOA between male and female mice. The serum level was in the range of highly exposed fluorochemical workers. We were able to calculate a serum to drinking water concentration ratio, which was approximately 19. This is significantly lower than the serum to drinking water ratio that's in humans, which is about 141. And these differences are result in differences in pharmacokinetics across the species. So that's why we had to have a significantly higher concentration of PFOA in the drinking water than a human would ever be exposed to in order to generate a human-like serum concentration. So onto the data. We were not surprised to find, and this is in our humanized PPARa alpha model that's fed an American diet, we were not surprised to find that PFOA increased liver to body weight ratios in both males and females. This is commonly observed. There was also a significant increase in liver triglycerides that was stimulated by PFOA treatment. And we see in the histological analyses that it's microvesicular steatosis that's occurring. So accumulation of small lipid droplets within the hepatocytes that's associated with this increase in serum triglycerides. Also notice that we see that PFOA induced a greater accumulation of triglycerides in the liver in the males than in the females. So our next question was the change in liver weight associated with the increase in triglycerides? And in the humanized PPARa, so here we see a correlation of triglyceride to liver weight in both females and in males. And you can see with increasing triglycerides, there was a corresponding increase in liver weight. And not surprisingly, there was a steeper slope, a greater amount of accumulation of triglycerides in the males than in the females. This effect does not occur in the livers of PPARa alpha null mice. PFOA did not increase liver triglycerides in mice that expressed no PPARa alpha. What you see here is a basic structure of a triglyceride. It's a glycerol molecule that's substituted with fatty acids, which are referred to as the building blocks of triglycerides. And currently we're investigating the specific types of triglycerides that PFOA is stimulating the accumulation of. So undoubtedly, PFOA is disrupting lipid homeostasis in the liver itself. But our next question was whether the serum lipids were similarly affected. So in these same mice, we did an analysis of the cholesterol particle profile and determined the concentration of cholesterol in each portion of the profile. So if we look here at females to begin with, so here you see that the vast majority of cholesterol is found in these HDL, high density lipoprotein cholesterol particles. There's almost nothing in the LDL range, and there's almost no change with treatment with PFOA. Something different happened in the males. So first of all, you'll see that, that males carry, albeit a small amount, but a larger amount of cholesterol and LDL particles than females, and that PFOA significantly increased the proportion of cholesterol being carried in LDL. So not only did PFOA increase total serum cholesterol in the humanized mice, that there was a shift in the distribution so that more cholesterol was being carried in the LDL particles. So I'm a molecular toxicologist, and I want to know what pathways could be contributing to these changes. And to begin to answer, to determine what changes are necessary to cause this impact on serum cholesterol, we identified 4 potential processes in the liver that could change serum cholesterol. 1, there could be an increase in cholesterol synthesis. There could be an increase in cholesterol export from the liver into the circulation. There could be decreased uptake of LDL cholesterol from the circulation into the liver, that reverse cholesterol transport. Or it was also possible that cholesterol was being inefficiently eliminated, inefficiently processed so that it could actually leave the body. So typically what we do when we start these investigations is we identify genes that we think of as biomarkers that are essential for each of these pathways. So what did we see? So again, these results are from our humanized mice. And I'll point out to begin with that the only significant changes that we saw were in the female mice. And that all of the significant changes that we observed were reductions in gene expression rather than increases in gene expression. So I'll point out that the gene HMGCR is responsible for making the protein, which is the rate-limiting step in cholesterol synthesis. This was significantly decreased by PFOA in female mice. The LDL receptor is the protein which is responsible for moving those LDL cholesterol particles out of the circulation and back into the liver. This was also significantly decreased in females by PFOA. Lastly, we have CYP7A1, which is a protein which is in the rate-limiting step in the synthesis of bile acids. So in order to get cholesterol out of the body, we change them into bile acids, and then those bile acids are stored in the gallbladder and released into the intestines to leave the body. And again, we have a very strong decrease in CYP7A1. In comparison with PPAR alpha null mice, it shows that these reductions of HMGCR and CYP7A1 are PPAR alpha dependent. But notice that the change in LDL receptor is not PPAR alpha dependent, that there is a significant sex difference here in response to females and males, and that our choice of biomarker genes does not yet explain the effect of PFOA in the males, in that PFOA significantly increased serum cholesterol. So we have more work to do here. So now let's look at what happened with serum triglycerides. And now we have another story emerging. So PFOA, first of all, if we look at the humanized mice, so these are the data, the serum triglyceride concentrations in female and male humanized PPAR alpha mice. So PFOA did not decrease serum triglycerides in female or male mice. What was even more intriguing, however, was that in the mice that did not express human PPAR alpha, PFOA significantly increased serum triglycerides. The same trend is there for males, it's just not as strong as in females. So we wanted to see whether the increase in serum triglycerides was associated with an increase in VLDL particles. So now we're looking at data only from female mice, and we're looking at the triglyceride profile across the different particle types in either humanized PPAR alpha mice or null mice. So you see in the humanized PPAR alpha mouse that's just had regular drinking water, there's very little triglyceride in the serum at all, and that there is a small increase in VLDL in the PFOA treated mice. The second thing to notice is that in the null mice, again, being fed this American diet, but just being treated with vehicle, there's an increase in very low density lipoprotein that is strongly increased in the PFOA treated mice. So again, we turn to analyze expression of genes that are biomarkers of processes involved in fatty acid and triglyceride homeostasis. So remember that triglycerides are these glycerol molecules with 3 fatty acids hooked up to them. The fatty acid pool, again, here's our liver. Fatty acid pool in the liver is determined by fatty acid uptake. So there are fat-free fatty acids that are traveling in your bloodstream, and these can be transported into the liver by this protein called CD36. The liver also has the capacity to make make fatty acids. So both fatty acid synthesis and fatty acid uptake determine the fatty acid pool. The fatty acids have to be activated in order to be hooked up to the glycerol molecule. So the next step is fatty acid activation, which should be apparently numbered number 3. Number 4 is that the activated fatty acids are sequentially added to the glycerol molecule, first to form a diacylglycerol, and then finally to form the triacylglycerol, which is also known as the triglyceride. Those triglycerides are then stored as fuel. They can be broken down to release fatty acids for oxidation, or then can be exported to the circulation as packaged in very low density lipoprotein particles. PPAR alpha is very well known for its regulation and induction of fatty acid release from triglycerides and their use to generate energy through the process of oxidation, which is necessary during fasting. But what I wanted to show you was the effect of PFOA on genes involved in triglyceride synthesis. And so what you can see is that across the board, PFOA, again, this is in our humanized PPAR alpha mice, that in both females and males, PFOA across the board typically increases the expression of CD36, which is necessary to take up fatty acids from the circulation. Fatty acid synthesis, which is carried out by this protein generated from the gene FASN. This was particularly upregulated to a small extent in males. ACSL5 is involved in fatty acid activation, a small but significant increase in fatty acid activation. The strongest increase was seen in the gene, one of the genes responsible for forming diacylglyceride, so adding that second fatty acid to the glycerol molecule. And then finally, there was a specific change, there was a specific increase in DGAT1, which catalyzes that final formation of the triglyceride, specifically in females. So comparison with the PPAR alpha null mice shows that induction of all of these genes is PPAR alpha dependent. So we had only one dose in our study, And we were curious about whether our results fit in with other results from the literature. And we found 2 other studies with similar treatment timing that also reported serum PFOA and triglyceride concentrations. The data are only from the male mice. So triglyceride concentrations in the male mice from our study is indicated by the hatched bar. The red arrow is at an occupational exposure, sort of maximum occupational exposure level. And what we can see is that serum triglycerides increase when serum PFOA levels are at or below occupational exposure levels. At higher non-human relevant levels, PFOA decreases serum triglycerides. So it appears that PFOA induces a non-monotonic effect on serum triglycerides. Non-monotonic dose responses are actually quite easy to explain once one realizes that they are simply 2 different dose response curves superimposed on one another. So for instance, at low concentrations, MIE, remember that molecular initiating event A, is activated, leading to an increase in response. At higher concentrations, a second distinct molecular initiating event, MIE-B, is activated, leading to a decrease in effect. So when the response or the effect is shown across the entire dose range, you see that non-monotonic dose response. Okay, so for a non-monotonic effect to occur, at least 2 molecular initiating events must be engaged. So is there evidence that PFAS activate more than 1 molecular initiating event? The answer is yes, and we see it even in our own mice from this particular study. So for instance, so here we have females on the top, males on the bottom, and we have both data from humanized mice and those that do not express PPAR alpha. ACOX is a target gene of PPAR alpha. So PFOA increases ACOX expression in mice that express human PPAR alpha, but it does not do so in mice that do not express PPAR alpha. That means PPAR alpha is necessary for PFOA to induce this gene. GS3MT is a target gene of another nuclear receptor called the constitutive androstenedione receptor. And what you see here is that in both PPAR alpha expressing mice and in mice that don't express PPAR alpha, PFOA still increases the expression of GSTM3. So in actuality, there are potentially multiple molecular initiating events for PFAS. So there's PPAR alpha. There are other nuclear receptors, the androgen receptor, the constitutive androstenedione receptor, estrogen receptor, pregnane X receptor, something called HNF4 alpha. PFOA can also bind to fatty acid binding proteins and fatty acid receptors. Okay, so in conclusion, you might have noticed it's complicated, but we're sorting it out. Some key takeaways are that PFOA at occupational exposure-related serum concentrations induces liver hypertriglyceridemia and induces serum hypercholesterolemia, particularly in male humanized PPAR alpha mice. PFOA at occupational exposure-related serum concentrations does not decrease serum triglycerides in male or female humanized mice, and in fact, PFOA induces serum hypertriglyceridemia, particularly in female PPAR alpha mice. The data are consistent with human epidemiological data, and they're consistent with a multiple PFAS, multiple MIE-driven non-monotonic dose response. And finally, developing human-relevant in vivo models will be essential to understanding mechanisms of action and biological effects of mixtures of PFAS, because the in vivo models can take into account those more in a more human-like manner in PFAS, or the odd chemistry of PFAS, as well as their effects across organs. And thank you very much.
Oh, great. Well, thank you so much, Jennifer. That was a lot. my brain
I
is
definitely
hurting.
went a little longer than I expected.
We do have time for a question or two. I don't see any that have come in. Someone did ask about the slides, and yes, the slides will be posted at some point in the next few days. It takes a little while. I can't do them myself, but they should be posted soon. So lots of just people confirming that this was very interesting and well done.
Great.
Any questions, anybody?
I can also, I'm also happy to take questions after Graylin has spoken in case someone becomes inspired.
Okay, well, we do have a couple of questions that have come in. One is,
Okay.
Have you looked at other PFAS besides the PFOA?
So we have not looked at other PFAS yet. Where we've been focused is actually repeating the PFOA experiment, but going lower in the dose response, because we actually think that lower in the dose response is where, number one, we become more and more human relevant, but we think that's where the balance of MIEs is actually going to reveal a quite strong effect on both increasing serum cholesterol and increasing serum triglycerides. But with that said, our next PFAS of interest is Gen X.
Okay. One of the more modern, shorter chains.
Right.
And another question is just, do PFAS compounds also interact with PPAR gamma?
Okay, so.
Or PPAR, sorry, not RA.
So no, PPAR gamma, right. So there's PPAR alpha, PPAR beta delta, and PPAR gamma. I would say at best, it's not a very potent or efficacious interaction. The activation of PPAR alpha is much stronger than PPAR gamma. So I'm not willing to completely rule out that PPAR gamma can and is activated, but the concentrations that are required and the low efficacy of the effect makes me question whether that would be a truly important mechanism of action.
Okay, and maybe we'll take one more, which is sort of a comment, but a question as well. It would be interesting to test various PFAS. Oh, now people are typing in their questions. Okay, let's see. Have you implemented human gene-modified mice studies on other pollutants? On your other pollutant studies?
No, we haven't. The human PPAR alpha mouse is the first humanized mouse model that we've worked on. What we do is work with human cell lines and primary human cells as a complement to these humanized mouse models. The humanized mouse models have been hard to come by, but now with genome editing technology coming along so far, such a long way, I think that there will be. I'm very interested in using some of the other humanized mouse models, but I haven't done them with the other chemicals.
Okay, and we'll just do this one last one and then move on. So you show that most cholesterol in mice is found in HDL rather than LDL, which is different than humans. Does this difference affect the interpretation of your results? Sorry if I missed that point in your presentation. So yeah.
So So certainly that argument has been used to negate the importance of results in mouse models. Basically in mice, the LDL is cleared really quickly. So we have a much harder time detecting changes in it. It's not that it's not happening. So first of all, our humanized mouse model actually, and which we're crossing to a black 6 strain, produces one of the most human-like cholesterol particle profiles. So number one. Number two, the biological pathways that contribute to cholesterol and triglyceride homeostasis are shared between mice and humans, with the exception of that CETP protein. So I think that the answer is that those changes that we're seeing in VLDL and LDL are human relevant.
Great. So we'll wrap up with that. And now I'm going to switch over to our second presentation, which is Graylin Nielsen, sorry about that, who in the Environmental Health Department at the Boston University School of Public Health. Her dissertation research focuses on molecular mechanisms that contribute to adverse effects of PFAS on liver function and leveraging high throughput in vitro data to predict downstream health outcomes. So with that, take it away.
Thank you so much for the introduction, Jennifer. And either Jennifer, if you could just let me know if you're seeing the wrong screen on my slides, that would be wonderful. And a huge thank you to NUMOA for hosting this series on PFAS and PFAS research. It's obviously a very important and emerging topic that we're exploring right now. And I'm going to be presenting about the modeling approaches that we use in our lab to predict the effects of chemical mixtures on molecular initiating events and how we're applying these approaches to mixtures of PFAS. I'm hoping to show you today that PFAS have complex interactions with molecular machinery, but that these complex interactions are not too complicated for us to predict with appropriate biological systems and modeling techniques. So we know that there are approximately 4,700 PFAS and PFAS mixtures with unique registrant numbers. And actually EPA lists over 9,000 unique chemistries on their PFAS master list on ComTox. PFAS are widely used in products that have direct contact with humans and the environment, for example, in fast food packaging, nonstick cookware, personal care products, and firefighting foam. And in the US, the largest producers of PFAS by volume are the electrical equipment and machinery manufacturing industries. The widespread use of PFAS means that humans are exposed to mixtures of these chemicals in food, water, air, household dust, soil, breast milk, and consumer products. Exposure to mixtures is documented in human biological samples. 4 PFAS are routinely found in more than 90% of the US general population. Individually, PFAS are associated with adverse health effects in human epidemiological studies, including decreased immune response, certain types of cancer, and elevated cholesterol levels. Most of what we know about the health effects of PFAS comes from studies with PFOA and PFAS, and now with increasing numbers of toxicology studies with PFHXS and PFNA. Because of the adverse health effects associated with PFAS exposure and physical properties that make them persistent and mobile in the environment, state, federal, and international bodies are increasingly regulating and issuing health advisories for PFAS in drinking water and other environmental media like soil, biosolids, air, food, and food packaging. PFAS guidelines, advisories, and regulations are based on health risk. In the case of drinking water health-based values, some are legally enforceable while others are not, but all are based on an assessment of health risk. Risk-based approaches are also driving regulations in other exposure sources, such as the tolerable weekly intake levels set for PFAS in food in Europe. In the US, the regulatory landscape is shifting rapidly. This map is just showing states that have drinking water guidelines that are either legally enforceable or not. And we are expecting to see action at the federal level on PFAS, likely in the form of an MCL, and potentially also listing PFAS as hazardous substances under CERCLA. For states that have drinking water guidelines and regulations, some states are choosing to set individual health-based values for each individual PFAS, while other states like Connecticut, Massachusetts, Vermont, and Washington are setting single values for multiple different PFAS, or regulating them as mixtures. It's important to remember where these health-based values come from. In the US, health-based values are derived from the reference dose paradigm. The point of departure for deriving reference doses comes from health effects research. Typically, animal toxicity studies will identify a dose that causes a sensitive health effect. That dose is then converted to a human equivalent dose and is divided by uncertainty factors to account for differing sensitivities between human populations and other sources of uncertainty. The resulting reference dose is a dose that sensitive populations can be exposed to daily without experiencing adverse health effects, specifically the adverse health effect that contributes to the point of departure. And in the world of molecular toxicology, we look at events occurring at the cellular level that can lead to the observed health effects in animals and humans that serve as the basis for these reference doses. And as Dr. Schlesinger mentioned in her presentation, the linkage between changes at the molecular level and subsequent health effects is known as the adverse outcome pathway. So this is how we conceptualize adverse outcome pathways. Starting all the way over on the left, we consider properties of the chemical and how it may interact with cellular machinery. The way that chemical affects changes in the cell is known as the molecular initiating event. And as Dr. Schlesinger covered, some examples of molecular initiating events include interactions with nuclear receptors in the cell. And the cellular response would be, for example, a change in gene expression, which can result in changes in protein levels within the organ, resulting in specific changes for reproductive systems or other organ systems in the body, and then an effect occurring at the population level. This AOP framework is helpful to integrate information and research at all levels of biological organization, and it can be used to inform risk assessment. So for example, by grouping chemicals that may share an adverse outcome pathway. Again, Dr. Schlesinger did show you an example of an AOP in how we think about the connection between PFAS, PPAR alpha, and changes in lipid levels. And this is just another example of an AOP for the estrogen receptor. The molecular initiating event here again is when a chemical binds to the estrogen receptor. This causes changes in gene expression and protein expression at the levels of cells expressing the estrogen receptor and reproductive tissues. And at the individual level, this can lead to altered reproductive behavior and function, and ultimately a reduction in population growth. So thinking back to the reference dose paradigm, where we're identifying a sensitive health effect in experimental animals, the AOP framework becomes helpful to see if we can identify the one or multiple critical molecular initiating events that lead to the development of that sensitive health effect. When we can identify a critical molecular initiating event leading to those sensitive health effects, we can then study the effects of chemicals on those key events and make predictions about health outcomes. And this is the approach that we're using in our lab to examine how environmental chemical mixtures affect key molecular initiating events, and then working forward to make predictions about health outcomes downstream. One of the key molecular initiating events that we study is nuclear receptor activity. Nuclear receptors are proteins that recognize and respond to molecules in the body like hormones, as well as therapeutic drugs and environmental chemicals. And here, recognize means that the molecules, therapeutic drugs or environmental chemicals will bind to the nuclear receptor. And respond means that the nuclear receptor will then bind to a region of DNA and affect changes in gene expression. So this is a diagram of what nuclear receptor activity looks like in cells. We have ligands like hormones produced in the body, drugs, environmental chemicals that come into the cell, bind to the nuclear receptor in the nucleus of the cell, and then nuclear receptors recognize specific regions of DNA called response elements. when the nuclear receptor is activated by these ligands, one effect can be that the genes downstream from the nuclear receptor response element are transcribed and ultimately expressed as proteins in the cell. Again, this is just one of multiple ways that nuclear receptors can change gene expression. This table is showing some examples of common nuclear receptors that we think about as molecular initiating events for environmental chemicals, but it is, of course, in no way exhaustive because there are numerous nuclear receptors in the body. So one example is the aryl hydrocarbon receptor, which is involved in cell determination and cell cycle regulation. We know that dioxin-like chemicals and halogenated and polycyclic aromatic hydrocarbons can operate through the aryl hydrocarbon receptor. And the estrogen receptor is another nuclear receptor involved in female reproductive biology, as well as other physiological functions, such as maintaining cardiovascular health. Bisphenols and organophosphate flame retardants are some examples of environmental chemicals that affect estrogen receptor activity. And another group of nuclear receptors is peroxisome proliferator activated receptors. There are 3 subtypes, including PPAR alpha, PPAR gamma, and PPAR beta delta. And these nuclear receptors play a critical role in fatty acid and lipid homeostasis, as Dr. Schlesinger covered in her presentation. Phthalates, organotins, organophosphate flame retardants, and PFAS are ligands of different subtypes of peroxisome proliferator activated receptors. This is the process we use in our lab to study the effects of chemicals on nuclear receptor activity. So we create a reporter construct, which has the response element for our nuclear receptor of interest, a reporter gene, which is typically a gene encoding for a fluorescent protein or other light-emitting protein. And we introduce this reporter construct along with our nuclear receptor of interest into the DNA of a cell that we can grow really easily in our lab. Using these cells with our reporter construct, we can then treat the cells with chemicals or chemical mixtures. If a chemical binds to the nuclear receptor will recognize the response element that we put into the cell's DNA and cause transcription of the fluorescent protein that we also put into the cell. The cell will then go ahead and make the fluorescent protein. And we can measure intensity of the light emitted from the cells to quantify the ability of chemicals to activate our receptors of interest. We analyze the results by comparing the intensity of emitted light to the intensity of a positive control chemical, which is a chemical we know fully activates our receptor of interest. And the data we produce are dose response curves, which show the relationship between increasing concentrations of the chemical we're testing and the activity of the receptor measured as emitted light. And we can use the properties of these dose response curves for individual chemicals to predict how a mixture would activate nuclear receptor activity. So there are 2 important properties of these dose response curves that are really important for how we model the effects of mixtures. And the first property is the maximum response or efficacy of the chemical. This is the maximum level of receptor activity a chemical causes when a dose response curve starts to level off. So we can see here, the red line is identifying where this dose response curve is leveling off, and that activity level on the y-axis is our maximum or efficacy. Chemicals that cause 100% of the maximum receptor activity are considered full agonists, and chemicals that cause less than 100% of the maximum activity are considered partial agonists for the receptor. We can compare chemicals by looking at the different efficacies. So here in this figure, we see that chemical A is a full agonist because it causes 100% of the maximum activity, and chemical A is more efficacious than chemicals B, C, and D, which have lower efficacy and would all be considered partial agonists. The other value that we use to quantify receptor activity is potency. And potency is the concentration of a chemical that is needed to induce 50% of the maximal activity. So we just had identified our maximal activity here. We find 50% of the maximal activity on the y-axis and can find the dose that causes that 50% activity level. More potent chemicals are chemicals that require lower doses of the chemical to elicit a 50% response of the maximum activity. So here we can see chemicals A, B, C, and D with their dose response curves. And for chemical A, the dose required to cause 50% of the activity is much lower than for B, C, and D. And we would say that chemical A is more potent than B, is more potent than C, is more potent than D. So these parameters are really important because we can use potency and efficacy from individual chemicals, again, to predict the effects of chemical mixtures on nuclear receptor activity using mathematical models. So two models that I'll be talking about today that we use to predict the effects of mixtures are relative potency and generalized concentration addition. Relative potency assumes that all chemicals in the mixture have the same maximum efficacy, but chemicals can have different potencies. And you may be familiar with the relative potency approach because it's the approach we take in risk assessment with some chemicals, for example, polycyclic aromatic hydrocarbons and some PCBs to quantify the effect of mixtures of those chemicals. And generalized concentration addition is another mathematical model that we use that considers both maximum and potency to make predictions of the chemical. So it allows chemicals to have, in the mixture, to have different potencies and different efficacies. We use these two modeling methods to predict the activity of chemical mixtures, and we've done this for multiple different systems. I'll report data on PPAR gamma, which again is one of the nuclear receptors that plays a critical role in lipid homeostasis. So we first validate the predictive capacity of our modeling approaches by treating cells with increasing doses of both a full and partial receptor agonist. And we display the experimental data here in 3D space. So for this experiment, we were using the full PPAR gamma agonist, which is rosiglitazone, a therapeutic drug. And you can see the dose response curve for rosiglitazone on PPAR gamma activity is here on the left. And the partial agonist that we used for PPAR gamma is NTZD-PA, which is a synthetic chemical. You can see the dose response curve for that chemical here. And we notice that it's a very, very low efficacy chemical because the maximum activity it causes here is considerably lower than for rosiglitazone, which is a full agonist. And the surface of the plot is what you observe when you treat cells with increasing concentrations of both NTZD-PA and rosiglitazone. So for example, this black line here is representing the dose response curve for NTZD-PA with a constant concentration of rosiglitazone. Similarly, this line here, this black one where my cursor is the dose response curve for rosiglitazone with a single concentration, this concentration of NTZD-PA. And this mixture's response surface is something that we observe in the cells that we treat and then compare to the predicted activity with our modeling approaches. So you can see that for relative potency, we're assuming that NTZD-PA and rosiglitazone both have the same activity level, the same maximum activity level, which means that we're actually over predicting the experimental data in the region where we would expect the curve to be limited by the minimal efficacy of NTZD-PA. With our GCA model, which accounts for both full and partial agonists, we can see that our surface is much closer to the experimental data because we're allowing this mixture to have the non-complete, the partial agonism of NTZD-PA. So we can conclude from this analysis that GCA is more accurately predicting the activity of mixtures of full and partial PPAR gamma agonists on PPAR gamma activity. We can then take the next step to use these modeling approaches to predict the effect of complex mixtures of environmental chemicals. So we tested a mixture of phthalates, including multiple different phthalates with differing efficacies and potencies. We make a mixture with fixed ratios of the individual phthalates, and then make dilutions of that mixture to come up with a dose response curve for the complete mixture. So now we're presenting on the x-axis is the concentration of the phthalate mixture, and again, the activity is presented on the y-axis. We know the concentration of individual phthalates in the mixture at each dose, and that's the concentration that we use to predict the effects of the mixture activity on PPAR gamma activation. So here we predicted the effects using relative potency and generalized concentration addition, in addition to a third approach called effect summation that I didn't mention earlier. So we can see the experimental data are shown with the black circles here. This is our actual mixtures data. GCA is the prediction shown here with the red curve, and we can see that generalized concentration addition is closely predicting the actual mixtures activity that we observe. Whereas for relative potency, which is assuming that all of the components in the mixture have the same maximum efficacy, we're over predicting the effects of the mixture on PPAR gamma activity. So we can conclude that GCA is providing more accurate predictions of the effects of multi-component phthalate mixtures on PPAR gamma activity. And though I won't show the full results here today, we've also found that GCA accurately predicts the effects of full and partial ligands of the aryl hydrocarbon receptor. And we also recently showed that GCA can be used not only for chemicals that activate receptors, but also for chemicals that antagonize nuclear receptor activity. So you may be wondering, we're here a couple minutes in, how does all of this fit into the PFAS picture with the PFAS mixtures that we observe in the environment? And the answer is we're exploring GCA as an approach to model the effects of PFAS mixtures on PPAR alpha activity. As Dr. Schlesinger introduced earlier, PPAR alpha is one of multiple key molecular initiating events that contribute to the adverse health effects associated with PFAS exposure. Our system for studying PPAR alpha activity uses the full-length human nuclear receptor in the same construct that I introduced you to before, where we're taking the nuclear receptor, response element, and fluorescent protein in the cell. And in this figure, you can see that PFOA, which is shown here in green, and GenX, which is shown here in navy, are both full agonists of PPAR alpha. They're causing approximately 100% activity here on the y-axis, while as the other PFAS that we have tested are partial agonists with efficacies between 15% for PFBS and 85% for PFNA. We also see that PFAS activate PPAR alpha with different potency, again, here with PFOA being the most potent of the chemicals, and PFBS here in gray being the least potent of the chemicals that we've tested. Because these differences in potency and efficacy, we think that GCA is an essential tool to predict the biological effects of PFAS mixtures. And we know that generalized concentration addition can provide more accurate predictions of PPAR alpha activity for full and partial PPAR alpha ligands. Here I am showing empirical and predictive effects for mixtures of full and partial PPAR alpha agonists. The full agonist that we use is the drug pemafibrate, and the partial agonist we use is mono-2-ethyl hexylphthalate. So here on this axis on the right, we have pemafibrate. We see the dose response curve for pemafibrate showing approximately maximal activity. And over here on this axis, we have MEHP. Increasing concentrations of MEHP cause a maximum effect of about 50%. So MEHP is a partial PPAR alpha agonist. And as we observe in our experimental data that the mixture surface, similar to PPAR gamma, has a variable response. And when we predict that response with relative potency, which again is assuming that in this case, both of the chemicals we test have the maximal activity of MEHP, we're actually under predicting the effect of the mixture because we're limiting the efficacy at that same level for MEHP. Whereas with generalized concentration addition, when we're allowing both efficacy and potency to vary, we capture a more accurate prediction of the surface of the mixtures that we're observing. And visually, it's relatively simple to see that, yes, this surface looks more like this surface than it does like this one, but we can also quantify that as well using the root mean squared error. This is just a measure of how different our predicted values are from our empirical values. So a larger value means that the predictions are further away from the empirical data. And we can see that for relative potency, it has a much higher RMSE than for generalized concentration addition. And because we can predict the effects of full and partial PPAR alpha agonists, we believe that we can apply this approach to complex PFAS mixtures to predict biological activity. So some key conclusions from our work thus far are that, one, we can use carefully designed human relevant biological systems to provide insight on the interaction between mixtures of environmental chemicals and key molecular initiating events like nuclear receptor activity. Modeling approaches like GCA that use information on potency and efficacy can accurately predict the effects of complex mixtures on nuclear receptor activity, including for PPARa. And PFAS are full and partial PPARa agonists, and mixtures will have complicated, but we anticipate predictable effects on PPARa activity. So this is just the first step of our research. We're using still a simplified system. And while these systems are very helpful, for example, in the high throughput setting where we can study high priority PFAS mixtures, for example, those commonly detected in human populations, though they're very beneficial in that high throughput setting, we also know that PFAS have multiple molecular interactions that will be more complicated than is predicted by this simple system. So as Dr. Schlesinger mentioned in her presentation, PPARa activation is one of several key molecular initiating events contributing to the health effects associated with PFAS exposure. So the next step of this research is to move from a simplified system into a more complex cell line which expresses other nuclear receptors and molecular machinery that PFAS can interact with. And with that, I would like to thank my research advisor, Dr. Schlesinger, as well as my BU collaborators, Dr. Thomas Webster and Dr. Wendy Hager-Bernays and Nathan Burritt. Thank you again to NUMOA for hosting this series of webinars about PFAS research and management. And thank you all for your time and attention. I'd be happy to take any questions.
Great. Well, thank you so much, Graylin. There have been a couple that have, well, a couple comments and questions that have come in. If people have any more for either Dr. Schlesinger or for Ms. Nelson, please let me know. Type them in. So one comment is just that Hawaii also has interim action levels. So they request that you update the map.
Oh, great. Thank you.
Knowledge that Hawaii also has some interim action levels. And then a question came in, which is relevant to me too, because I might have missed it, but is there an animal model as well that for your fluorescent reporter so that you could study in vivo effects as well?
So you can do a similar construct where you have a fluorescent protein expressed in an animal, but it's more common to use reporter genes. So for example, genes that we know are downstream from these receptors to record activity, because it's pretty simple to look at changes in gene expression. So when we do this in an in vivo system, we're actually looking at gene expression as our reporter construct, if you will.
Okay. Could you please confirm that the GCA model at the end, did you graph each of those individually, or was that the mixture response?
So the GCA data that I was showing with the mixture surfaces, that's very similar to what we do with PPARa gamma, where we're looking at the dose response information for 2 chemicals at a time, and the cells are treated with increasing concentrations of both chemicals. And let me see if I can go back to that actually. Yeah, so
I'll make one point that the relative potency and generalized concentration addition models, all they use to predict the surface are the individual dose responses of each of the chemicals. And that's what makes GCA so powerful, because all we need are all we need are dose responses for single chemicals that we can plug into, once we've confirmed that GCA accurately predicts responses of mixtures, then all we need is the dose response of individual chemicals, which are much simpler to generate than empirical dose response surfaces.
Yeah, and I think perhaps part of the question too was about the experimental design. So we have here again the increasing doses of pemafibrate, which means that each one of these lines is the pemafibrate dose response curve with a fixed concentration of our partial agonist. And again, here this is the dose response curve for MEHP with a single concentration of pemafibrate. So that's how we're generating that mixture surface. And then based on those concentrations, we can predict what we would expect the activity to be from those 2 chemicals.
Okay, here's some more questions coming in. We always seem to get back to, there's so many PFASs out there. People are looking at how to look at them as a whole class. So do you think GCA will allow us to establish a single cleanup level for PFAS as a class, similar to how we sort of have it for PCBs now? We kind of look at PCBs in total.
Yeah, so it can certainly contribute to that type of designation, again, because we're looking at one molecular initiating event and considering how mixtures would influence that. I would say I'm cautious with that statement because we're still a pretty far ways away from that. But that is our hope, is, we know that PFAS are going to have complex interactions, whether we're looking at PPARa alpha as our key molecular initiating event or the constitutive androstenedione receptor. But with generalized concentration addition, we can really account for some of that complexity. So we're hoping that it will be a useful tool for grouping PFAS ultimately.
Okay, I think this is a little related. So trying to envision the GCA method, because it looked like from the graphs you showed, there's just like 2 chemicals. So how about if you're doing 6 or 8 chemicals at the same time? How does that work?
Yeah, so we use these 2 chemical systems to validate that GCA and relative potency are behaving as we expect them to. And then the beauty is, for additional chemicals, we don't have to do studies with 1 chemical at a time. Once we know that the system is operating as we expect, we can then use the parameters from as many PFAS as we want in the mixture, or as many phthalates as we want in the mixture, to come up with a prediction based on the concentrations that we know are in our mixture, and that's how we compare it to the empirical data. But once we've made that assessment that it is working as we expect it to, then we can feel more confident in using GCA to predict the effects of more complicated mixtures.
Okay. So another comment that the state of New York has also adopted the drinking water MCLs for PFOA and PFOS, so I guess, which are 10 PPT each. They did that in August. So just again, I guess it's tough to keep those maps,
like I said, rapidly changing
to date, because things keep changing everywhere. Yeah, all the time. Okay, here's somebody that looks like they work in this field. So asking a very specific question. Do the PFAS stimulated changes in reporter expression change when cells are cultured in different matrices? For example, advanced organotopic or 3D systems versus 2D monoplayers. I don't know what that means, but hopefully you do, and others on the webinar.
Yeah, I don't know. I'm working for now in just a couple different cell systems, so I haven't tested something that's more complex and more similar to like an organ, for example. I don't know, Jennifer, if you worked with that, but certainly there are important experimental considerations that change how PFAS behave. So we worked long and hard to establish a system that gave us, one, the best response, but also seemed the most human relevant. That was some of the considerations for using a human nuclear receptor. We also have thought carefully about the type of serum that we're treating the cells with, because we know that PFAS bind to serum albumin. And there are certainly a number of considerations for experimental design that will have a bit of an influence on the result.
So the question is a really important one, and we are approaching, our approach is to start in as simple and as controllable a system as possible, which is these, this relatively artificial transfected COS reporter assay. Our next step is actually to go to, like in the case of PPARa alpha, to a human hepatocyte cell line. It took a long time to find the right cell line, the one that was most human-like. And what we use there, rather than a reporter assay, is endogenous gene expression. Could that be influenced by, culturing the hepatocytes in 3D? Probably, but I think that our goal is actually to move from reporter assay to endogenous, endogenous gene expression in a human cell line to a physiological endpoint in that human cell line to see how far can we go from the molecular initiating event and still be able to predict the effect of the mixture. But I think even better than, let's say, organotypic or 3D culture is then taking our model in vivo, which is going to have all of the, quote unquote, bells and whistles of pharmacokinetics, chemical transport, appropriate, nuclear receptor expression and activity. So it's a really important point. We need to get as in vivo and human relevant as possible. So that's actually what we're working towards, is doing the mixture studies in vivo. The Holy Grail would be able to develop a dose response in a cell culture model that predicts what happens in vivo, so we don't have to do in vivo anymore.
Yes, and you can, you can do it so much more quickly with so many different combinations and things as well.
Right, and with Graylin, while she wanted to, could not show her, erasing in culture. We can do something called array design to generate empirical data from complex mixtures. And so Graylin's actually done mixtures of PFAS up to 7 or 8 chemicals already, right, Graylin?
Yep.
Yeah.
Mm-hmm.
Okay, here's a related question again. When you have multiple chemicals, not just the 2, could competitive bindings change the results?
So competitive binding absolutely changes the results. And that is what I assume what they mean by competitive binding is the 2 different PFAS, for instance, both trying to bind to PPAR alpha. That is why GCA is so important, because perhaps you'll recall from the figure, some PFAS only modestly activate PPAR alpha activity, where PFOA strongly activates PPAR alpha activity. And so when those less efficacious chemicals represent a greater proportion of the mixture as a whole, mass balance will dictate that the receptor is going to be occupied much more likely with the less efficacious chemical. And that's what pulls down the activity of the mixture as a whole. So competitive binding and our being able to account for that competitive binding is what makes GCA really a step forward from the traditional additivity models.
Great. Let's see. Does anybody else have something to ask? We're coming up on 3 o'clock, but we probably have a minute for something else if anybody has anything. But I think we went through the questions that came in. So I don't see any more. So I really want to thank Graylin Nelson and Dr. Jennifer Schlesinger from the Boston University School of Public Health for presenting on their research, and we look forward to learning about the results of your work in the future. So I will repeat that the slides, the PDF of these slides will be posted on the NMO website, and when they are posted, I will send an email out to everybody that was on the webinar today to let you know about that. And the contact information for everybody, I believe, is included in their slides. So if you have follow-up questions, you can contact the presenters directly. So I'm just seeing more thank you and well done comments coming in. So with that, I think we will end today. Hope everybody enjoys the rest of their afternoon. And thank you once again, Graylin and Jennifer. And NMOA will have more webinars. They're on the NMOA website, and probably everybody today will end up getting an email in the next week or 2 about the upcoming webinars as well. So thank you again, everybody. Enjoy the rest of your afternoon. Bye-bye.